blood composition. We will study the components of blood and the function of each type of blood cell. In the human body, blood is the most important connective tissue. It is 5.5 liters of life force cruising through thousands of arteries and veins. These blood vessels carry blood to every other organ, tissue and cell of the body to keep them functioning and thriving. As we look inside an artery, we observe numerous blood cells. The red blood corpuscles are the most commonly found blood cells, and the number ranges from 4.5 to 5 million per cubic millimeter. This 5 liters of blood contains 25 trillion of these cells. The RBCs are spongy biconcave discs which have a diameter of around 8 microns. These RBCs are devoid of a nucleus, hence they are said to be enucleate. They consist of a sponge-like protein frame in which the hemoglobin molecules are present. The RBC absorbs oxygen on reaching the lung region and turns bright red in color. These RBCs travel within the blood vessels. On reaching the oxygen-deficient tissue cells, oxygen diffuses out of the RBC into the tissue cells. Here, the RBC becomes deoxygenated. Simultaneously, carbon dioxide from the tissue cells is absorbed into the RBC. The average lifespan of an RBC is 120 days, after which it disintegrates. Present in the blood vessels is another major component of the blood, and that is the white blood cells or WBCs. These cells do not have any specific shape. Their number ranges from 4,000 to 10,000 per cubic millimeter. They are of two main types. One type senses the microbe, attacks it, and engulfs it by phagocytosis. While on the other hand, the second type becomes activated on contact with a microbe. Then it starts to release specific antibodies which track down the microbe and adhere to it, making it immobile. Finally, the WBC engulfs the microbe, helping the body to resist infection. The average lifespan of the WBC is only 18 to 36 hours, and then they disintegrate. The third type of blood cells is the platelets, which are the smallest of the three. Their number varies between 0.15 to 0.45 million per cubic millimeter. These platelets have an average lifespan of 9 to 10 days, and then they disintegrate. Platelets are also known as thrombocytes. These cells adhere at the site of a wound, where they release blood clotting factors. This leads to the formation of thread-like structures which form a mesh-like network, thus preventing the escape of blood. The red color of the blood is due to the presence of RBCs. Along with these, other cells are suspended in the liquid component of the blood called plasma. This can be verified by collecting a sample of blood by a syringe. The collected blood looks red but is an aggregate of these components. To see all the components, we place the collected blood sample into a test tube, which is then placed within the centrifuge. The machine is switched on. A closer look at the contents of the test tube shows that the heavier RBCs start sinking to the bottom due to the spinning. However, the lighter WBCs and platelets swirl in the plasma above the RBCs. After centrifugation, the tube shows distinct layers of the components present in it. The topmost part is a layer of clear supernatant liquid, which is the plasma. The precipitate consists of the WBCs, platelets and RBCs. Recap. Blood is one of the most important connective tissues in the body. It consists of enucleated RBCs, which have hemoglobin molecules present in them. The RBCs absorb oxygen and provide them to the oxygen-deficient tissue cells throughout the body. WBCs are the other cells present in blood, which protect the body from attacking microbes by engulfing them. The third type of cells present in blood are the platelets, which come into action at the site of a wound. They form a clot and prevent the loss of blood from the wound. All these cells are suspended in the plasma present in the blood and can be easily separated into distinguishing layers by centrifugation.